In this video, we're going to talk a little bit about respiratory control. Right? So literally the control of breathing. And one of the first things to probably remember or to recall is that, if I, again, I'll draw very schematic lungs here. The trachea and bronchi, right? We have our lungs. Not even going to put the heart in here. Right? And those lungs sit within the thoracic cavity. Right? So I'm going to draw in black here this wall that sits around it. Right? The thoracic cage, if you will, right? Made of ribs and muscles. And then I'm going to fill in, actually, at the bottom, a layer of muscle that sort of sits here and it's kind of dome shaped. Right? This sort of thicker red wall at the bottom I'm drawing here is the diaphragm. Right? And so one of the things we have to recall is that ventilation occurs not because of muscular action within the lungs, but because of muscular action of the diaphragm. Right, label that guy here. That's our primary respiratory muscle. And then muscles in the wall or surrounding the wall of the thoracic cage. Right, so this is the thoracic cage or thoracic cavity wall. Right, we'll talk a little bit about the muscles involved there outside of the diaphragm in just a moment. So the key here is that to control respiration, we're not really controlling the lungs per se. We're controlling the muscles of the diaphragm and the thoracic wall or associated with the thoracic wall. And that's really important, right? Because that's we actually talked about this fluid-filled space, right? Remember this fluid-filled space? There's a fluid in here. I won't make it go all the way around. Right? But there's a fluid between the thoracic wall and the surface of the lung, right? That intrapleural fluid, or just pleural fluid for short. And that was produced by the serous membranes, right? The visceral and parietal layers of the pleural membrane. And one of the things it does is it creates surface tension between the lung and the thoracic wall. And because the lung is elastic, it actually has a negative pressure to it. So it tends to sort of pull the lung up against the thoracic wall. And that's really, really important because if we're going to control ventilation by changing the thoracic cage volume and not the lungs themselves, we need the lungs to stick to that thoracic cage, right? To have their volumes track the change in thoracic volumes. Right? So again, we're going to change thoracic volumes to change lung volumes, but that pleural membrane and the pleural fluid are critical to that linkage between the two. Right? The lungs have to stick to the inside of the thoracic cavity. Otherwise, you can change thoracic volume all day long, and the lungs would just sit there. Right? And that's actually called atelectasis, or a sort of the lay term would be a collapsed lung. Right, where the thoracic volume can change, but the lung is not attached to it, so it just sits there and remains unventilated, right? And that's obviously really bad. Okay, so the second thing that really we remember to point out here is that, again, referring to Boyle's law, is that it's the pressure gradient between P alveolus and P atmosphere. In other words, the pressure outside the lungs, P atmosphere, and the pressure inside the lungs, P alveolar or intrapleural pressure. The difference between them is what drives airflow, right? We get an air pressure gradient that drives flow into and out of the lungs. Again, go back to the video on Boyle's law and the mechanics of ventilation. Okay, well, why do we bring that up now? Well, one, it's related to what we're talking about here. It's changing thoracic volume. But the second reason is there's really two forms of breathing. And it's really a gray scale. It's a broader range. We're going to sort of point to two versions of it, but they're sort of the two extremes. Or you could think of one as anything other than the first. The first one is called eupnea, or eupneic breathing. And that's where that's breathing at rest. And to breathe at rest, we need a very small, again, so the point there is that we're moving small volumes. Tidal volume is that air that moves with each breath. And at rest, you're not moving an enormous volume, right? And so we don't need a very big pressure difference between the atmosphere and the inside of the lungs. And so when we generate small pressure differences, we're doing eupneic breathing. That's what we're doing at rest. However, when we become more active or more have an increased metabolism for whatever reason, we're going to need to move larger volumes of air with each breath, and that is called hyperpnea. And we usually refer to that as forced breathing. Meaning, we're going to be moving larger volumes. To move larger volumes of air with each breath, we need a larger gradient between the atmosphere and the intrapleural pressure, right? And so we're gonna have to basically employ more muscles to change the thoracic volume, essentially more dramatically, more rapidly, to generate that bigger pressure difference. A bigger pressure difference means more rapid air movement, right? Some of the worst weather we experience is when we have a very low atmospheric pressure, right? You'll get this strange weather event where the atmospheric pressure falls really low. Well, it falls really low relative to the surrounding areas. And so guess what happens? You have a big gradient. So what do you get? Wind, right? Because of the large gradient, the large difference between the high pressure areas and the low pressure areas. Okay, well, this is the same is true here. We're just actively generating a bigger difference between outside and inside by more rapidly changing the thoracic volume. And again, we'll talk a little bit about the muscles involved in that. And again, I'm not overly concerned that you plug in all of the anatomy here with the respiratory muscles, but I will point out the muscles that are doing eupneic breathing and the muscles that are doing hyperpneic breathing. Okay, so where is our control center? It's in the medulla. Right, that's where we find the respiratory control centers. Now, you will notice in your readings that there are respiratory control centers also found in the pons. When I'm referring to the ones in the medulla, I'm talking about the pacemaker control centers. These are the centers that contain the neurons that innervate the muscles that I was just talking about. So there are these regions in the medulla that have neurons that exit, right, and innervate the muscles like the diaphragm, right? So we can have let's draw a neuron here. Coming from, just sort of schematically, coming from the medulla and innervating, right, the diaphragm, because that's the location of the neurons that literally, and again, it might not be a single neuron here, I'm exaggerating, but the neurons connected to other neurons that are innervating the respiratory muscles, right? Okay, there's not one respiratory control center, there's actually three, and they are the DRG, that stands for the dorsal respiratory group. There is one referred to as the VRG, which is the ventral respiratory group. And then there's the one that I use the shorthand PBC. And that stands for pre-Botzinger complex. Right? So the DRG contains largely inspiratory neurons. Right? Its function is to stimulate the muscles that drive inspiration. And so that will, and that actually tends to be largely, the target tends to be diaphragm, which is going to mean something in a minute here. It means something else besides inspiration. right? And I should point that out. When the diaphragm contracts, it pulls downward, right? When it contracts, it pulls this way. And by doing so, it makes the thoracic cavity taller, if you will. It's sort of like a quasi-cylinder, if you will. And so if you think of a cylinder getting taller, it would have a larger volume. If it has going back to Boyle's law, if it has a larger volume, then it has a lower pressure. And that way, if we lower the pressure inside relative to outside, air would move in, right? And of course, right after that, we would do the exact opposite and we would move air back out again, right? But the DRG stimulates the inspiratory centers, it is an inspiratory center that stimulates the diaphragm to increase the thoracic volume and drive inspiration. Okay, the VRG is both inspiratory and expiratory. So both inspiratory and expiratory. And that's a little strange, right? Because that's two different sets of muscles. It can't be the same muscles, right? And it is, again, we'll explain more in a moment. And it is active mostly during hyperpnea. Right? And remember, hyperpnea is this forced breathing. And when we have forced breathing, we have to more dramatically change the thoracic volume, which means we need accessory respiratory muscles. In other words, it's not just the diaphragm anymore. And again, that would be just inspiratory. And so there are accessory muscles that drive inspiration. In that group, we would include things like the sternocleidomastoid, the scalenes, the external intercostals, and all of these, just lump those together. Sternocleidomastoid, scalenes, and external intercostals. What do? They pull the ribs up. By pulling the ribs up, they make the thoracic cage, because your ribs kind of tend to hang down, right, toward the front. They sort of droop. When we pull them up, it makes you more round in cross section. Take a deep breath, right? Your rib cage, right? Your ribs kind of come out and up, right? And so what that means is that you become a little bit more cylindrical, which is going to increase the volume. Increasing the volume decreases the pressure. Decreasing the pressure drives inspiration, right? And so those were the inspiratory accessory muscles. The expiratory accessory muscles basically sort of squash your thoracic cavity, right? So think internal intercostals and your the abdominal muscles, all layers of the abdominal muscles, the obliques and the transversus abdominis and the rectus abdominis. They all tend to sort of squash your belly when you contract them. And what that does is it pushes your diaphragm up. If it pushes your diaphragm up, it decreases volume. If it decreases volume, it increases pressure. If it increases pressure, it drives air out. That's why those are expiratory muscles, right? So for hyperpnea, we need to more actively increase the thoracic volume by pulling up on the ribs, right? Sternocleidomastoids, scalenes, external intercostals. And for more dramatic expiration, we have to either pull the ribs down, the internal intercostals, or push the diaphragm up, the abdominal muscles. Okay? And so the VRG is going to be all about driving air movement when we need larger volumes, when we need to enhance that pressure gradient that drives air movement. Okay, well, we got to bring in this guy. What is this guy doing? The pre-Botzinger complex is thought to be, and this is not well elucidated, thought to be the primary pacemaker. And when we say that, what we mean is that there are neurons in this medullary control center that spontaneously fire. They will generate a rhythm. Now, that's not to say that that rhythm can't be modified. We will point out in just a moment that there is input that can, think about it, right? Does your respiratory rate and depth change when you exercise? Of course it does, right? And so you can't just have a pacemaker that decides that, your respiratory rate should be 15 per minute and leave it at that for the rest of your life, right? You wouldn't be a very good tennis player. And so we've got this pacemaker region. And again, all these areas, show pacemaker function. Again, it's very complex, but the pre-Botzinger complex is thought to be sort of the primary pacemaker. And so this guy is helping to drive the rate of the DRG and, if needed, the VRG. Okay. Now, let's come back to that. What is the input, right? So again, we can a good way to think about this, right? You learned about the SA node in the heart. It's an automatic pacemaker. It does not require any input whatsoever, and it will generate a heart rate of about, 70 to 85 beats per minute. Yet it has input, or there are input two centers that will then generate output that can modify that heart rate, as well as contractility, right? And so we have the same potential here. We can alter the rate or the strength of the thoracic wall, muscular changes to change the volume that we're moving, right? We can both breathe faster and we can move more air with each breath, but we need to know when that needs to occur, right? And so we need to have input here, and that input falls largely in two categories. And in this case, the input, these are the integrating centers. The medulla is the integrating center. So it receives this input and then sort of, knows what that input means and if an adjustment should be made to solve the initial problem. And so what kind of input? Well, we have chemoreceptors and we have stretch receptors. And by stretch receptors, I mean stretch receptors that are within the lung wall. Right? The chemoreceptors sense, and you should recall this, we've talked about this previously. The chemoreceptors sense, I'll squeeze it in up here, or I can actually draw an arrow because this is what the chemoreceptors would actually measure. They measure pH as H+, right? pH is a measure of the concentration of H+, PCO2, or the partial pressure of carbon dioxide, and PO2, the amount of oxygen. And what you might find interesting is that we would think, breathing, it should be controlled by the amount of oxygen we have in our blood, right, or in our brain tissues, right? And that's true, but remember, breathing serves multiple functions, right? Breathing not only brings oxygen in, but it expels CO2. And CO2 is linked to H+ via the carbonic anhydrase, right? The carbonic acid equation, the carbonic anhydrase is the enzyme. And so, right, as CO2 goes up, so too does H+, right? It's automatic because of the hydration of CO2, and that reaction will result in the release of free protons. So every time, a good way to think about this is when, and again, just as an example, exercise. When you exercise, right, you would be short on oxygen initially, right, until the respiratory system can respond. So O2 would go down. The cells are using more energy, so CO2 would go up. Because the CO2 is going up, the H+ would go up, right? And remember, H+ and pH are inversely related. So if H+ goes up, then pH actually goes down. Okay, that's the log scale for pH, inversely proportional to concentration of H+. Okay, where are these chemoreceptors? Let me make some room here so I can squeeze them in underneath here. Right? They are found in the brain and the large arteries, mainly the aorta and what's called the carotid sinus. And the carotid sinus is simply this right where the common carotid artery splits. There's this sort of bulge, right, where it splits into the internal and external carotid. And right at that location, we have this cluster of chemoreceptors. right? So in the wall of the aorta, in the wall of the carotid artery, and in the brain tissue itself, actually very closely adjacent to the medullary centers, we have these chemoreceptors that are sensing the blood chemistry, right? Or in the case of the brain, the interstitial fluid chemistry of the brain, right? And again, if brain, the chemical balance of the brain, O2 and CO2 and pH are off, we need to solve that problem quite rapidly. Are you ready for this? These receptors respond so dramatically that when you breathe in, your blood chemistry changes slightly. In other words, in that heartbeat that carries blood away from the lungs, the output of the chemoreceptors changes based upon each heartbeat, right? And so it's a very rapid, right? It's going up and going down based upon the extraction of oxygen and the removal of carbon dioxide with essentially each pulsation of the heart and increased perfusion of the tissues. It's very rapid responding receptors. Okay. All right, so we've got our pacemaker driving in, and we've got our input from the chemoreceptors. Oh, we need our input from our stretch receptors. I'll explain this one too. These are actually called the Herring-Brewer reflexes. Herring-Brewer reflexes. And these simply are stretch receptors in the wall of the lung that indicate both when the lung is becoming well inflated or when it's becoming underinflated. So for instance, if you're inhaling, the stretch receptors that are responsive to increased inhalation are going to start to increase their firing. They're going to start to tell the brain, hey, your lungs are becoming full. And so what it's going to do is it's going to tell the inspiratory centers to shut off so that we switch over from inspiration to expiration. When the lung volume, and then you start to expire, right? The lung volume goes down. And when the lung volume gets low, there are a separate set of receptors that start to say, hey, lung volume's getting low, you need to breathe in. And so it'll turn back on the inspiratory center and turn off, if it's on, the expiratory center. Right? And so it helps us transition between inspiration and expiration with each breath based upon, the sort of the local, are the lungs, filling or, deflating at a given moment. Okay. The last thing I wanted to do, actually, there's one other thing I want to point out. There's this region, I mentioned this in the beginning, there's a region in the pons that is part of respiratory control. We call these pontine centers because they're located in the pons. And these two centers do not directly control the respiratory muscles. There's one called the apneustic center. And then there's one called the pneumotaxic center. Right? And what these guys do is they get some of the same input from chemoreceptors and stretch receptors. And what they do is they modify the output of the medullary receptors. And so what does the apneustic center does? What does it do? It stimulates inspiration. Right? So this is the center that's tending to drive larger inspiratory volumes. Right? When people get defects in their respiratory control centers after brain traumas and things like that, sometimes they can end up with what's called apneustic breathing, where they breathe in sort of excessively, this really long inhalation before they will breathe out. Okay, so that is stimulating or having input largely to the DRG, right? Because it stimulates inspiration. Look at the DRG, right? It's driving inspiration. The pneumotaxic, it does just the exact opposite. So it's going to shut off inspiration and help initiate expiration. And so what these two centers are doing is basically helping us control sort of the volume of each breath by sort of controlling the transitions between inspiration and expiration and vice versa. Okay? So again, those centers are more controlling, having input to the centers that are actually doing the stimulation of, and again, that's largely these two. Right? Remember the pre-Botzinger, it does have neurons that control muscles, but it's largely thought to be the pacemaker. But your two centers that are really focused on only the muscular control would be the DRG and VRG. Okay, great. So the last thing I want to do is help you put it into context with, like, back up here, like with what is being active during eupnea versus hyperpnea. Okay? And to do that, I want to clear the boards for a second. And this hopefully will bring things sort of full circle. Right? And so there's really, if we sort of lay out what the possibilities are here in terms of breathing cycles, we have eupnic inspiration, right? So that would be breathing in at rest. We have eupnic expiration or exhalation, same thing, right? Breathing out at rest. We have hyperpnic inspiration. That would be breathing in a forceful manner. And then we have, and I'm abbreviating here obviously, hyperpnic expiration, which would be breathing out forcefully. Okay? Now, let's draw a little grid here. You can start to see how these fit in. Right, so let's put our centers up here. We have our DRG. And the DRG only contains inspiratory neurons, right? We have the VRG, and it has both inspiratory neurons, so the little I here for inspiratory portions of the VRG. And we have the VRG expiratory neurons. So we're going to put a little E down here. All right, we'll make our columns. Make our rows. Okay. All right, now all we need to do is go across and see which centers are active when. Well, eupnic inhalation is breathing in at rest, right? And that, the DRG contains only inspiratory neurons. It's eupnic, so we don't need the VRG. Remember, the VRG is for hyperpnea only. So we already know that that's not going to be active here at all, right? So active only, the VRG is the only center active, or sorry, the DRG is the only center active during inspiration, eupnic inspiration, right? Eupnic expiration. It's not the DRG, right? Because the DRG is inspiratory. It's not the VRG inspiratory, and we're certainly not doing forced expiration, right? Because remember, it's eupnea. So it's nothing, nothing, and nothing. And what that means is that exhaling at rest is passive. In other words, it does not involve muscular contraction. It's all elastic recoil of the lung and gravity. Right? So we're not using any muscles to exhale at rest. Now let's go to an exercising scenario, and we're doing hyperpnic breathing. And so now hyperpnic inspiration. Well, the DRG contains inspiratory neurons, so they're on. But the VRG controls the accessory muscles of inspiration. And remember, if we're doing hyperpnic breathing, we need the accessory. We need the sternocleidomastoid and the scalenes and the external intercostals, right, to pull up on those ribs. So that guy's active. And of course, the expiratory neurons and muscles don't need to be active during inspiration. Okay, and then hyperpnic expiration. Of course, the DRG is off. The inspiratory VRG neurons, they certainly wouldn't play a role. And so we're going to be using only the expiratory neurons of the VRG. And again, This is where we have to add muscles. Remember, it's passive at rest. Now we have to add muscles to sort of squash that thoracic volume. So the internal intercostals and the abdominals that helps sort of shove that diaphragm upward, reduce the volume of the thoracic cavity, and drive that air out. Okay, and that sums up the roles of the DRG and VRG during both eupnic. Right? Remember, this is at rest. And hyperpnic. Right? Remember, this is forced. And again, you could do that intentionally by hyperventilation, but of course it's what's gonna happen during exercise when you get that increased both rate and depth of breathing. Hyperpnea. Okay, that's everything.